Ready? In previous class, you already discussed about the plant breeding process and also discussed a new high yielding varieties in wheat, rice, sugarcane, bells, etc. by the plant breeding process. And also discuss about traditional plant breeding and conventional plant breeding. With alpha this plant breeding, the plants are resistant to different types of disease and pest resistance, disease resistance and pathogenic resistance, etc. etc. Now in this class I want to discuss about another important essay question concept. That one is tissue culture. In the plant breeding, the technique of tissue culture has emerged a very efficient tool. That one is the tissue culture is a very efficient tool in the plant breeding program to prograde between improvable crops. What type of crops? Improvable crops like disease resistance, pest resistance. Plants are improvable with help of the a very efficient tool in the plant breeding process. That one is tissue culture. Okay, the tissue culture much approachable technique than the other traditional plant breeding technique. Other traditional plant breeding techniques to poliste, this tissue culture is very efficient technique. Understand? Very efficient technique. Next one. In this tissue culture, by using this method, cells, tissues, embryos, etc. etc. use as a explant. Okay, in this tissue culture, by using this method, cells of the root, the cells of the stem, cells of the leaf, cells of the apical buds, etc. and the tissues, different types of tissues, and the embryos. See, it consists of embryos in their stature. Thus, embryos or tissues or cells used as a material, the main material to culture in this tissue culture technique. Okay, and this maintain and grow in vitro under aseptic conditions, not septic condition. It is aseptic condition. These cells or tissues or embryos are maintained grow in vitro under aseptic conditions in artificial method, artificial medium, extensively used MS medium in tissue culture. And this the nutrient medium providing necessary nutrients for growth. Tissues gani, cells gani, embryos gani grow avalante some nutrients are necessary for their growth. So we are prepared the new artificial medium to providing necessary nutrients for the growth of tissues, for the growth of cells, for the growth of embryos. So this nutrient medium is available in the nutrient necessary nutrients providing in the nutrient medium culture. That one is known as culture medium. Culture medium. This culture medium is prepared with help of the necessary nutrients. Some necessary nutrients are provided in the culture medium. These nutrients are helpful for to grow cells, tissues, embryos into entire plant. Understand? In this way, the previous experiment is done on the tissue culture by several scientists. Like suppose in 1839, Sweden and Swan, there are also expand. The cell is the basic unit of life. Cell is the basic unit of life and it capable of autonomy, self-replicable. So the cell is self-replicable and to forms a new cells by the division. Okay, this phenomenon is known as autogamy. The autogamy capability and also basic unit of life is cell, noted by the Sweden and Swan. Okay. ఈచ్ సెల్ క్యాపబుల్ ఆఫ్ ఏ హోల్ ప్లాంట్. ఒక సెల్ నుంచి కంప్లీట్ ప్లాంట్ ని తయారు చేయొచ్చు బికాస్ ఆఫ్ ద ఆటోనమీ. ఓకే, నెక్స్ట్ వన్. In 1902, G. Herbernot, okay, he was introduced the in vitro culture method. But in this in vitro culture method, different types of plant pots are utilized as explant, but he may not prepare the whole plant from the explant. So his experiment was failed. Okay, the callus culture is failed. Understand? So, but the callus in vitro culture method is introduced by this G. Herbernot. So he was considered as a father of tissue culture. Who is the father of tissue culture? G. Herbernot. Understand? In 1907, the Herbernot experiment was re-experimented by the Henning. He was experimented done on cruciferous embryos of crucifers. He was successfully grow on the explant of the cruciferous embryo because of the he was successfully obtained the maturity of the plant from the embryos of crucifers. ఓకే, క్యాబాజీ ఫ్లవర్ ఉంటుంది కదా, ఆ క్యాబాజీ ఫ్లవర్ లో ఉన్న క్యాబాజీలో ఉన్నటువంటి ఎంబ్రియోస్ తీసుకుని ఇట్ వాస్ ప్రిపేర్డ్ టు మెచ్యూరిటీ, వాటి నుంచి ఒక మెచ్యూరిటీ ప్లాంట్ ని తయారు చేశారు. ఓకే, హూ ఇస్ సైంటిస్ట్? హెన్నింగ్ ఇన్ 1907. Sister Simon in 1908. In 1908, Simon produced callus. He was successfully produced callus from birds, roots, popular stem segments. He was completely prepared callus from these pods. Okay. Next one, Simon experiment is mainly based on callus culture. He was utilized callus culture. It was based on callus culture and also successfully produced callus from the birds and callus from the roots and callus from the popular stem segments, etc. Okay. Next one, in 1934, experiments on tomato roots. He was experimented on tomato roots and also prepared a successfully entire whole plant from the root pods. Understand? Some important technical terms in the tissue culture. That one is explant and totipotency. What is explant and what is totipotency? Basically, any part of the plant grow in a nutrient medium. Any part of the plant grow in a nutrient media under sterilized condition gives rise to a new whole plant. Whole new plant is formed from any part of the plant. Growing in nutrient media under sterilized condition. Sterilized condition లో nutrient medium మీద any part of the plant body, లీఫ్ తీసుకుంటే లీఫ్ కానీ స్టెమ్ కానీ రూట్ కానీ ఎనీ పార్ట్ ఆఫ్ ద ప్లాంట్ ఒక ఎక్స్ప్లాంట్ గా తీసుకుంటే దాని న్యూట్రియన్ మీడియం స్టెరిలైజ్ కండిషన్ లో గ్రో చేస్తే ఇట్ గివ్స్ రైజ్ టు ఏ హోల్ న్యూ ప్లాంట్, ఒక కొత్త న్యూ ప్లాంట్ అని ఫార్మ్ చేస్తుంది. ఓకే, the step of any part of the plant are known as explant. ప్లాంట్లో ఏ పార్ట్ని అయితే మనం తీసుకుని కల్చర్ చేస్తున్నామో ఆ పార్ట్ని, సపోజ్ లీఫ్ని తీసుకుంటే లీఫ్ ఈజ్ ఎక్స్ప్లాంట్, స్టెమ్ పార్ట్ని తీసుకుంటే స్టెమ్ ఈజ్ ఎక్స్ప్లాంట్. ఓకే, రూట్లో ఒక పార్ట్ని తీసుకుంటే దట్ పార్ట్ ఈజ్ ఎక్స్ప్లాంట్. ఒక ఎంబ్రియోని తీసుకుంటే దట్ వన్ ఈజ్ ఎక్స్ప్లాంట్. Okay, in this way, any part of the plant growing in nutrient medium under sterilized condition gives to a whole new plant. That parts are known as explant. Those parts are known as explant. Okay. Next one, the capability. That means a entire whole new plant is formed from the any part of the plant. That means the parts of the plant parts are capability to form a entire whole new plant. This type of capability is known as totipotency. What is totipotency? Totipotency means nothing but the entire whole plant is formed from the any part of the plant body. This type of plant body parts capability is known as totipotency. Understand? This totipotency is require nutrient medium. This nutrient medium contains sucrose, inorganic salts, vitamins, proteins, and also growth regulators. Okay, the nutrient medium is mainly consist sucrose, inorganic salts, vitamins, proteins, growth regulators. Growth regulators means such as auxins and cytokinins. Okay, in this way the nutrient medium under sterilized condition, any part of the plant body gives rise to a whole new plant. That parts are known as explant. Thus capability is known as totipotency. The sterilized nutrient medium is consist sucrose, inorganic salts, vitamins, proteins, growth regulators like auxins and cytokinins. Let's discuss about the tissue culture techniques. It is very important and also 8 marks question this one. Listen carefully. Okay, what are the steps are involved in the tissue culture techniques? The first one, preparation of culture medium. We are prepared culture medium. Okay, this culture medium consists of several valuable nutrients or carbon sources. What are the types of carbon sources? What type of carbon sources? Sucrose and inorganic salts, vitamins, amino acids, and the growth regulators like auxins and cytokinins, etc. consist this culture medium. So you are prepared initially culture medium. Okay, this culture medium consists high nutrient. So some microbial organisms can grow in this culture medium, nutrient medium. Okay, because of this reason the nutrient medium is contamination occurs. So this contaminated culture medium, hence it is sterilized, must be sterilized. Your sterilized culture medium. Okay, preparation of culture medium and also sterilization of culture medium is essential for the tissue culture program. Second step is sterilization of medium, nutrient medium. In this the stream of sterilization is called autoclave. Autoclave is essential for the sterilization of the medium. Next one, the medium is autoclaved for 15 minutes at 121 degrees centigrade and also 15 pound pressure is available in the autoclave. So your autoclave or sterilize the medium with help of the autoclave. Once see the autoclave diagram. Autoclave is essential for the sterilization of the nutrient medium. Next one, inoculation of explant. Initially, your explant, this one is the agar plant. This agar plant, any part of the plant pot is used as explant. In this, the leaf is extracted from the agar plant, so the leaf is a explant. This leaf is sterilized or cleaning with help of the fresh water. Okay, you are cleaning the surface of the leaf. Then after, the leaf is also sterilized. Then this leaf is cut into small fragments or small pods. Okay, these small pods are known as explant. This explant is inoculated into the nutrient medium under sterilized condition. Under sterilized conditions are available in the laminar airflow instrument. Okay, this laminar airflow instrument is consist of one base and also alcohol flame, and also some instruments are helpful for inoculated explant into the test tube sterilized medium. So you are inoculated explant into the sterilized medium under sterilized condition. These under sterilized conditions are available. One of the instrument, that one is laminar airflow. Once say this laminar airflow. Inoculation means that the explant is transferred to sterilized medium under sterilized condition. That one is known as inoculation. Okay. After inoculation, these test tubes are incubated. Okay. Incubation of explant. So these after inoculation into the explant is inoculated into the medium. This entire test tubes are incubated up to 3 to 4 weeks. 3 to 4 weeks. 3 నుంచి 4 వారాలు దాన్ని ఇంక్యుబేట్ చేస్తాం. ఓకే, you are using incubated chamber. Look at this. Incubated chamber, you are incubated 3 to 4 weeks after inoculation of the explant in the nutrient medium. Okay, after 3 to 4 weeks, we are appear some mass of tissues. That means nothing but after incubation of 3 to 4 weeks, you are observe some mass of tissue. It is a irregular, unorganized and undifferentiated mass of actively dividing cells. There are undifferentiated mass of tissue and also actively divided cells. That undifferentiated mass of tissue is known as callus. Okay, the callus is formed. In sometimes this callus is differentiated into some organs like root and shoots. In this way, the organogenesis is occurred by the presence of auxins and cytokinins are present in the nutrient medium. Okay, in this way, organogenesis occur. That means the undifferentiated mass of cells is differentiated into organs. That one is known as organogenesis. Organogenesis. Okay, organogenesis. Sometimes embryo-like structures are also visible after incubation period. Understand? After incubation period, some embryo-like structures are appear on the culture medium, nutrient medium. So this one is known as embryogenesis. Okay, explant nunchi manaki embryos form aithe, that one is known as embryogenesis. The type of embryos are known as embryoids. Understand? Embryoids. So the after incubation, the explant is directly formed callus, or directly formed organogenesis, or directly formed embryo-like structures. That one is embryogenesis. These embryos are known as embryoids. Not embryos, embryoids. Okay. In this way, the member of embryos, or member of organogenesis-like structures, member of mass of tissues are formed from the explant. Understand? So within a short time, member of plants are produced. In tissue culture, perform using a small part of a plant to develop a large number of plants. Large number of plants in a short period. చాలా తక్కువ టైంలో ఎక్కువ ప్లాంట్స్ అనేవి ఒక పార్ట్స్ నుంచి, ఒక సెల్స్ నుంచి మనకి ఫామ్ అవుతుంది. దిస్ టైప్ ఆఫ్ phenomenon is known as micropropagation. Micropropagation means nothing but within a short time a number of plants are produced from the explant is known as micropropagation. So the micropropagation, member of plantlets are produced from the same plant explant. So these are genetically, morphologically and genetically equal to the parental plant. So these all plants are micropropagationally formed a numerous plant. These all plants are known as soma clones. Understand? Soma clones means nothing but all the plants are developed of the same part of the plant, and also these are all genetically identical. So these are called soma clones. Okay. Some meristems are also transferred to pathogens due to the lack of after the formation of organogenesis or embryoids, entire plants are, after formation of callus, this callus is transferred into auxin and cytokinin containing, that means a plant growth regulator containing medium. Okay, the callus is cleavage into different types of different parts of tissues, and these callus parts are transferred into plant growth regulator containing nutrient medium. What are the plant growth regulator? Auxin and cytokinin containing nutrient medium. Hence, by the presence of auxins and cytokinins, this callus is differentiated into plantlets. Understand? In this way, the plantlets are, after a callus, these test tubes are contained plant growth regulators. These callus are differentiated into plantlets, a small plant-like structures. These are called plantlets. After formation of these plants, these plantlets are transferred into pots. ఒక కుందీలోకి మార్చుకోవాలి. And then after, these pots are transferred into glass house. Okay, they transfer into glass house. That means the plantlets generated through organogenesis or embryogenesis need to be acclimatized before they are transferred to pots. Understand? Pots. ఓకే, ఈ విధంగా పార్ట్స్లోకి మార్చే ముందు మనం వీటిని ఏం చేస్తామంటే ఒక గ్లాస్ హౌస్లోనికి మార్చడం జరుగుతుంది. Okay. గ్లాస్ హౌస్లోకి మార్చిన తర్వాత దేర్ ఆర్ ట్రాన్స్ఫర్ టు ఫీల్డ్. దట్ మీన్స్ గ్లాస్ హౌస్లోకి మార్చిన తర్వాత వీటిని ఏం చేస్తారంటే జాగ్రత్తగా మళ్ళీ ఫీల్డ్లోనికి ఎంటర్ చేస్తారు. గ్లాస్ హౌస్లో సక్సెస్ఫుల్గా గ్రో అవుతాయి, ఎందుకంటే మన స్టెరిలైజ్డ్ కండిషన్లో ఒక అబ్జర్వేషన్ కండిషన్లో ఉంచుతాం కాబట్టి. దెన్ ఆఫ్టర్ వీటిని మళ్ళీ తిరిగి ఫీల్డ్లోకి ఎంటర్ చేయాలి. ఫీల్డ్లోకి ఎంటర్ చేసేటప్పుడు సాయిల్కి అడాప్ట్ అవ్వాలి. సాయిల్కి మెల్లిమెల్లిగా గ్లాస్ హౌస్లో అడాప్ట్ చేస్తుంటాం కాబట్టి దెర్ ఆర్ ట్రాన్స్ఫర్ ఇన్ టు డైరెక్ట్లీ ఫీల్డ్. ఫీల్డ్లోకి ట్రాన్స్ఫర్ చేసిన తర్వాత దెర్ ఆర్ సక్సెస్ఫుల్లీ గ్రో ఇన్ టు న్యూ ప్లాంట్. అండర్స్టాండ్? Advantages of the tissue culture. Some advantages of the tissue cultures are: first one, a large number of plants can be grow in a short period of time. That one is micro propagation. Okay, చాలా ఎక్కువ నెంబర్ ఆఫ్ ప్లాంట్స్ ని చాలా తక్కువ టైంలో మనం ఫామ్ చేయొచ్చు. ద నెక్స్ట్ అడ్వాంటేజ్ ఇస్ హెల్దీ ప్లాంట్స్ కెన్ బి గ్రో ఫ్రమ్ ద డిసీజ్ ప్లాంట్. సపోజ్ ఒక ప్లాంట్కి డిసీజ్ వచ్చింది అనుకోండి, ఆ డిసీజ్ ప్లాంట్ లో ఎపికల్ మెరిస్టమ్స్ ఆర్ లాటరల్ మెరిస్టమ్, ఎపికల్ అండ్ లాటరల్ మెరిస్టమ్ మనం తీసుకుంటే అప్పటికి అవి టిష్యూస్ ని అంటే జైలం అండ్ ఫ్లోయం టిష్యూస్ ని ఫామ్ చేయవు కాబట్టి దేర్ ఆర్ ఫ్రీ టు ద వైరల్ డిసీజెస్. ద స్పాట్స్ ఆర్ ఫ్రీ టు ద వైరల్ డిసీజెస్. సో ఆ పార్ట్స్ మనం తీసుకుని ఒక ప్లాంట్ ని తయారు చేయొచ్చు. ఆ ప్లాంట్ అనేది డిసీజ్ ఉండదు. సో హెల్దీ ప్లాంట్ కెన్ బి గ్రో ఫ్రమ్ ద డిసీజ్ ప్లాంట్. డిసీజ్ ప్లాంట్ నుంచి ఒక హెల్దీ ప్లాంట్ ని గ్రో చేయొచ్చు. నెక్స్ట్ వన్, ప్లాంట్స్ వితౌట్ సీడ్స్ కెన్ బి మల్టిప్లైడ్. ఒకవేళ సీడ్స్ లేకపోయినా కానీ వాటిని మల్టిప్లై చేయొచ్చు. ప్లాంట్స్ వితౌట్ సీడ్స్ కెన్ బి మల్టిప్లైడ్. అనదర్ అడ్వాంటేజెస్ ఇస్ ఇట్ ఈస్ యూజ్ఫుల్ వేర్ సెక్సువల్ రిప్రొడక్షన్ ఈజ్ నాట్ పాసిబుల్. కొన్ని ప్లాంట్స్ లో సెక్సువల్ రిప్రొడక్షన్ ఉండదు. అటువంటి సెక్సువల్ రిప్రొడక్షన్ లేనటువంటి ప్లాంట్స్ లో నుంచి కూడా మనం ఏం తయారు చేయొచ్చు? ప్లాంట్స్ ని తయారు చేయొచ్చు. Understand? These are the main advantages of the tissue culture. Understand?
In plant breeding, the technique of tissue culture has emerged as a very efficient tool to propagate better improved crops. Tissue culture is much approachable technique than other traditional plant breeding techniques. By using this method, cells, tissues, etc. are maintained and grown in vitro under aseptic conditions in artificial medium, providing necessary nutrients for growth. In 1839, Schleiden and Schwann proposed that cell is the basic unit of life and is capable of autonomy. Each cell, if given an environment, is capable to regenerate into a whole plant. Based on this theory, a German physiologist, Gottlieb Heberland, in 1902 developed the concept of in vitro cell culture, also called tissue culture. Cells in his experiment increased in size but failed to divide, due to which his predictions failed. However, his hypothesis paved the way for more research. He is thus regarded as the father of tissue culture. Taking cue from his experiments, Hanning in 1907 took nearly mature embryos of crucifers and successfully grew them to maturity. In 1908, Simon regenerated callus, buds, and roots from poplar stem segments and established the basis for callus culture. A breakthrough came from White in 1934, who experimented on tomato roots. He reported not only could culture tomato root tips grow, but they could be repeatedly subcultured to fresh medium of inorganic salts supplemented with yeast extract. During 1950s, scientists found that any part of a plant grown in nutrient media under sterile conditions can give rise to a whole new plant. The plant part used in culture is called as explant. This property of plant cells to give rise to a whole new plant is called totipotency. The term totipotency was coined by Stewart in 1968. Totipotency. It requires a nutrient medium that contains sucrose, inorganic salts, vitamins, proteins, and growth regulators, such as auxins and cytokinins. Technique of tissue culture. The first step of tissue culture is excising or cutting a small part of plant tissue, like the leaf, root, or shoot of a plant. This cut-out part, explant, is then placed in a suitable nutrient medium containing a carbon source such as sucrose, inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. The explants are allowed to multiply and form a group of tissues called callus. Callus is irregular, unorganized, and undifferentiated mass of actively dividing cells. This callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth. Tissue culture is performed using a small part of a plant to develop a large number of plants in a short duration. It is thus also called as micropropagation. As all these plants are developed from the same part of the parent plant, they are genetically identical and called soma clones. Micropropagation is extensively used in horticulture and floriculture and is employed in the large-scale production of tomato, banana, apple, etc. This technique can also be used to propagate high-yielding disease-resistant varieties in a short span. Even in conditions when a plant is diseased, tissue culture is used for recovering healthy plants from diseased plants by taking the explant of the apical or axillary meristem. This is done because the meristem is always free from pathogens due to lack of vascular supply. The process of using the meristem for propagation is known as meristem culture. Culturing of meristems has been successfully carried out in plants such as banana, sugarcane, and potato. There are many advantages of tissue culture, which are as follows. A large number of plants can be grown in a short period of time. Healthy plants can be grown from diseased plants. Plants without seeds can be multiplied. It is useful where sexual reproduction is not possible.